Hello, folks. We are going to be talking about meiosis today. All right. So, meiosis is how we get to sexual reproduction. So, sexual reproduction combines two types of cells, your gametes, eggs from females and sperm from males, to create a fertilized zygote. Or fertilized egg, which then becomes a zygote. By mixing the chromosomes from two individuals, we have increased genetic diversity as opposed to mitotic reproduction, where you're basically just cloning individuals. So the process of meiosis is very similar to the process of mitosis, except we have two stages here. So most plants and animals have two sets of chromosomes, one from mom, one from dad. This means that they are diploid. Di means two or 2N. So like body cells are 2N. They have the full complement of chromosomes needed to make another skin cell, for example. However, we have to avoid doubling of chromosomes when we're talking about sexual reproduction. So our reproductive structures that produce gametes, our testes or our ovaries, are going to have cells that only have a single set of chromosomes, because remember, meiosis is combining our gametes, which is our eggs and our sperm. And so we wouldn't want eggs and sperm to have the full complement of DNA that they need, because one, then we can't combine between individuals, and two, we'd have way too many chromosomes. So our gametes are going to be haploid or 1N. Okay, so our mitosis is going to again be mostly for body cells, and in some organisms, some organisms actually reproduce via mitosis, via parthenogenesis or simultaneous, as in Komodo dragons do parthenogenesis, and simultaneous hermaphrodism occurs in both the mangrove killifish and clownfish. So this is where the simultaneous hermaphrodites undergo meiosis inside of their bodies, because they have both tissues from testes and ovaries in their bodies, and then they reproduce mitotically that one version of meiosis. Whereas meiosis itself is going to have two phases, one where the chromosomes split once, and then another where the chromosomes split again. So we have meiosis 1 and meiosis 2. So during meiosis 1, okay, prophase 1 is super important. And this is where we get some of our genetic variation, where we have what's called crossing over of chromosomes, which is where the chromosomes sort of like dance around and touch each other. And when they touch each other, they trade pieces of DNA with each other. So it's really cool. This is how we get our genetic variation. And this is why, for example, not everyone in the same room getting COVID would respond to COVID the same, because we all have different genes that help confer us different genetic variability and different immune systems. So anyways, back to the actual process. So meiosis 1, prophase 1, a big deal, crossing over. Then we have metaphase 1, where the chromosomes align at the metaphase plate. Anaphase 1, the chromosomes are pulled apart. And telophase 1, we have cytokinesis. So this is really very similar to mitosis, with the exception of crossing over in prophase 1. Now, again, talking about what happens with meiosis 1, we have chromosome replication in S phase makes identical sister chromatids, and they're going to remain attached at the centromere. Okay, so in prophase 1, we have our homologous pairs of chromosomes, so one from mom, one from dad, and this is where they touch each other and trade segments of DNA. Okay, so this is exactly what happens with crossing over, where this is where we end up with different types of genes in the same places on the chromosome. So meiosis 2 basically looks, again, very similar to mitosis. It's almost identical to mitosis, because this is where we're actually splitting those sister chromatids apart into individual chromatids. So we actually, and there is In the process of chromosome alignment between meiosis 1 and meiosis 2, it differs, where we have, and prometaphase 1 and anaphase, we are pulling these pairs of homologous chromosomes, whereas in prometaphase 2 and anaphase 2, we're pulling apart sister chromatids. So we're going from 4 to 2, and then from 2 to 1. So each phase of meiosis reduces the chromosome number, okay? And then again, anaphase, telophase, and cytokinesis are just like they were in mitosis. I love this.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
Okay, so I love that animation. It's great to study. When we compare meiosis to mitosis, okay, both both processes divide the nuclei. However, mitosis divides the nuclei once, meiosis divides the nuclei twice. Homologous chromosomes pair to form tetrads in meiosis. Remember that was those 4 chromosomes, as opposed to mitosis that only has those 2. Mitosis produces 2 final cells, meiosis with a full complement of DNA, where meiosis produces 4 total cells with a half complement of DNA. In meiosis, we have crossing over between the sister chromatids when they touch each other and exchange genetic material, which helps us with genetic variation. And then meiosis itself reduces chromosome numbers from 2N to 1N, and mitosis does not. So here's a look at a comparison between mitosis and meiosis. So you can really see that meiosis 2 is almost identical to mitosis, whereas meiosis 1, we have that crossing over in prophase 1. All right, thank you so much for your time. Have a wonderful day.